Hi, my name is Andrea Navarrete Vargas, and my science story begins in 1999. I was born in Veracruz, Mexico, and for the first 3 years of my life, I was surrounded by lots of family, lots of food, and lots of love. After moving to the United States, I was continually reminded of the many influences my early childhood environment had on who I am and who I developed to be as a person. My research with Dr. Laura Borodinsky at UC Davis has allowed me to apply my experiences with environmental influence on the nervous system. For my research on how the environment influences spinal cord development has been extra special to me. Here we see the embryo of an African clawed frog going through neurulation, the formation of the spinal cord.
Motor neurons serve as highways that send messages from the spinal cord to the muscle. Some of these messages include flexing, contracting, and overall muscle movement. Unfortunately, in disease, the death of these motor neurons lead to severe defects due to an inability to initiate or control muscle movement. The most common disease that results from motor neuron degeneration in adults is amyotrophic lateral sclerosis, or ALS. Despite the importance of having neurons during development, as much as half of our neurons will go through programmed cell death, a process also known as apoptosis. However, this pruning of neurons by cell death is normal during development. Our bodies know how important neurons are, so much so that during development, they will produce more neurons than we need in order to ensure that if anything in our environment changes or a neuron doesn't make the proper connections, we have a backup. Specifically, I look at how temperature influences the development of spinal cord motor neurons. My goal is to understand how motor neuron survival increases at cold temperature by investigating and identifying the molecules responsible for protecting motor neurons from cell death during development.
Here we have a transverse section of spinal cord. Previous work in our lab found that animals grown at cold temperature exhibited a higher number of motor neurons, as shown on the left, than animals grown at warm temperature, as shown on the right. They found that this increase in motor neuron number at cold temperature correlated with a decrease in apoptosis. Tryptamate is a cold-sensitive ion channel, permeable to calcium. Therefore, my hypothesis is that tryptamate activity is necessary for motor neuron survival in Xenopus laevis embryos raised at cold temperature. To test this hypothesis, fertilized embryos were injected at the 2 to 4 cell stage with a tryptamate morpholino or a control morpholino. A morpholino is an antisense oligonucleotide construct that blocks the translation of a protein. These embryos were then grown at 16 degrees Celsius or 22.5 degrees Celsius until they reached stage 40, which is when they were fixed and prepared for immunohistochemistry staining. Apoptotic cells were stained using the tunnel assay. Motor neurons were stained with an antibody for the HB9 transcription factor. On the left, we see the results of this staining for embryos raised at cold temperature. The graphs on the right show that embryos deficient in tryptamate had an increase in apoptotic cells and a decrease in motor neuron number. Additionally, we found that tryptamate knockdown led to an increase in apoptotic motor neurons. Specifically, 48% of motor neurons were apoptotic in the group deficient in tryptamate. At warm temperature, we found that unilateral tryptamate knockdown had no effect on the number of apoptotic cells in the spinal cord. This result is expected as tryptamate is not active at this temperature. This data allowed us to conclude that tryptamate knockdown and lack of tryptamate activation by cold temperature accelerates the rate of motor neuron programmed cell death. Additionally, that tryptamate function is necessary for the prevention of apoptosis of spinal cord neurons, specifically motor neurons in animals raised at cold temperature. I would like to thank Dr. Borodinsky and the rest of my lab, as well as the MARC program, for allowing me to conduct this research.